Okay, this is a quick revision video on the origins of humans, and we're going to cover some of the genetic evidence of human evolution, and in covering that we'll cover some basic genetics and look at genetic variation. So there's a whole series of lectures that I gave on human evolution, but all of these focus around the point that we're looking at where humans came from and the genetic evidence that says that humans came from a common ancestor of chimps and then a common ancestor of all mammals and so on. But we're really interested in the more recent genetic changes that have made humans humans. So we're going to cover the genetic evidence, looking at our similarities to chimps, similarities to other mammals. We're going to look at the rate of mutation and accumulation of genetic damage, and that genetic damage defines the genetic changes that are present in humans, but let's say not chimps. And then we're going to look at mitochondrial versus Y chromosome haplotype in later lectures, and look at how we can use this sort of genetic information to map population movements. So just as a quick overview, we can see that humans and chimps separated from each other relatively recently in the last 10 million years. Although humans didn't separate from chimps, we separated from chimps via a common ancestor, which was here. And then there was a common ancestor of great apes here, which is where the split between human chimps, gorillas, which also appear, and the orangutans. And then we can see a common ancestor of our group, the primates, with other primates and so on. So this gives you a scale in millions of years about how far back we are going. What we know is that at this point here, there was a split between the chimpanzee group and some of the other great apes, which have 48 chromosomes, and humans, which have 46 chromosomes. And those 46 chromosomes in humans, that is present in all of the human groups, so that's Neanderthals, Denisovans, and Homo sapiens. So this is summarised here on this plot. So we've got 10 million years here. Separation from a common ancestor with gorillas, which had 48 chromosomes, is about 9 million years ago. Separation from a common ancestor with chimps. That group retained 48 chromosomes. We have got 46 chromosomes. Then about 3 million years ago, we've got this particular sort of subspecies of primitive hominid about 3.5 million years ago in Africa. Then up here we've got several lines of hominids leaving Africa. So 1 to 2 million years ago we know hominids left Africa and colonized Europe, mostly Asia. They seem to have given rise to Neanderthals, Denisovans, which lived in Asia and Europe during that time. And then subsequently we've got Homo sapiens leaving Africa 130,000 years ago, and then a second wave 80,000 years ago, which we think populated Asia and Europe and subsequently Australasia. So we think that basically the population of Europe, Asia, Australasia came from this 80,000 year ago wave of migration out of Africa. Since Homo sapiens left Africa, let's say 80,000 years ago, they then came into contact with previous species or subspecies of Homo sapiens which were already living in Asia and Europe. So these are the Denisovans, particularly in Asia, and Neanderthals in Europe, but also in Asia. And what this illustrates is the amount of movement of DNA due to interbreeding between these different hominid species. So what we can see is the African population who didn't leave Africa have got no Neanderthal or Denisovan DNA within them. So if you go to sub-Saharan Africa, you find very, very little Neanderthal or Denisovan DNA. Whereas if you look at Europe, Asia and Australasia, you see that there is, particularly in Europe, there's quite a bit of Neanderthal DNA. In areas of Asia, particularly this Denisovan DNA, and what we know that these populations were interbreeding with the waves of Homo sapiens that moved into the areas where they were living and had been living there for hundreds of thousands of years. So what this shows is the movement of Homo sapiens through Southeast Asia, and what we think is that 60,000 or so years ago populations moved through into Australia, and these picked up Denisovan DNA, and what we can see in Australian populations and Papua New Guinea is quite high levels of Denisovan DNA within them relative to other populations which tend not to have picked up this DNA. We can see just from this map here where the first Denisovans were identified, which is in Central Asia. So the reason we know this is that we've identified DNA from a cave here of this ancient hominid species, which we call Denisovans, and then we look for markers in the DNA from the bones here, and look for markers which are unique to Denisovans and not present in modern Homo sapiens or Neanderthals, and we can say, okay, this is a genetic marker for this population, and then we look for those populations in look for DNA markers associated with these populations in other populations from around the world, and we find the highest concentration of genetic markers relating to this population, the Denisovans, in native Australians and Papua New Guinea and some of the Polynesian islands, suggesting that a wave of migration came through this region here. Interbreeding occurred between modern humans leaving Africa 80,000 years ago and the existing Denisovan population that were living in this region here, and then that DNA is being carried down this route down to Australia. And this allows us to track populations as they migrated tens of thousands of years ago. So we know that Neanderthal and Denisovan genetic signatures exist in modern humans but not in most Africans. And we know that Denisovan DNA exists most commonly in Southeast Asia, so that suggests that this population were present as modern humans migrated through that area. Now we know all of this because DNA mutates at a fairly constant rate. We know that populations that are separated will accumulate mutations independently of each other. So if a mutation occurs, let's say in the Denisovans, and they've been living isolated from other humans for 500,000 years, those mutations in the Denisovans should be fairly easy to identify from modern humans, and then you can track how much Denisovan DNA is present. Now when we look at some genes, we find some genes mutate very quickly, some genes mutate very slowly, and we can use these almost like a molecular clock for evolution studies. Some genes, particularly the histones, which you've heard about in the previous lecture, and Hox genes, which I mentioned very briefly in the previous lecture, these mutate very, very slowly because they're absolutely fundamental to how the organisms are put together. Others mutate much more quickly, and then non-coding DNA mutates quicker still. When I say mutates quicker, it all mutates at the same rate, it's just that non-coding DNA mutations are left in the genome, whereas mutations in Hox genes and histones tend to result in death of the organism, so therefore mutations are very rarely carried forward to the next generation. This is just showing the frequency of histone mutations between different species. So you can see humans, mouse, rat, cow, chimp, and this is the amino acid sequence, not the DNA sequence. So we're only really interested in mutations that affect coding sequence for this plot. And what you can see is that humans and chimps are the most closely related, and you can see that we've got the same amino acid here and here, whereas all the others have got a different amino acid. Same there, same there. Then at this particular place you can see that humans and chimps are the same, cow, rat and mouse are all different from each other. So that gives you some idea as to what is the order of events of evolution. and just based on this change alone, you would guess that humans and chimps and cows are more closely related to each other as a group than mouse and rat. And then from this change, you would suggest that humans, chimps, cows and rats are more closely related to each other, and mouse is sort of a bit of an outlier. Now, sometimes these can be a little bit misleading, in that mouse and rat are actually very closely related to each other. But when you build up changes that have occurred under over thousands of genes, you can build up a very definite picture of the order of events in evolution. Now, these are the Hox genes. I've briefly mentioned these in the past. This is a bunch of genes which regulate body segments and how they differentiate in embryogenesis, and these mutate at a very, very slow rate. What we find is that in the key regions of these genes, or the coding sequence, there's no amino acid changes between us and chimpanzees, or indeed mouse. So if you see human amino acid sequence there and the mouse amino acid sequence there, there's absolutely no change between us and mouse, because this is a very highly conserved gene. Even when you look at between humans and fruit fly, there's not an awful lot of change because it's such a fundamental gene. In other genes, which are not quite so fundamental or not even coding sequences, we find mutations occur much more quickly. And this is just one example that I talked about, which is a gene called the NANOG-P8 gene, which is not a proper gene really. And what we find is that if you look at the chimpanzee chromosome 15, there's this sequence here which matches exactly the same to the human chromosome 15, with the exception that there's a couple of thousand bases of DNA that have been slotted in. And that couple of thousand bases of DNA is what we call the NANOG-P8 gene. And this is just one recent, so almost like newly created gene that has occurred since our split between humans and chimps. So no chimps have got this NANOG-P8 gene. And just skipping forward a few slides, this NANOG-P8 gene exists, and it's a copy of the ancestral NANOG gene. And we think this gene emerged about 1 million years ago, and NANOG-P8 sequences have been found in Neanderthals. So that suggests that prior to our split with Neanderthals, the NANOG-P8 gene was present in those previous populations. So with both humans and Neanderthals who've got the NANOG-P8 gene, but chimpanzees don't have it. So we know that that is an event that has happened since our split with the common ancestor of chimpanzees. And it's data like this, thousands of different locations throughout the genome, that has allowed us to follow the order of events of evolution. In a later lecture, I'll talk about how we use things like SNP chips and whole genome sequencing to come to even more accurate predictions of the order of events.